Hey there, seventh graders. If you were in school on Tuesday and you need to finish up your lab for the plant and animal cells, um, just a few things. If you did not get done with all of the picture drawings, on the second half of the screen here, I have a document that I also attached to the assignment that's got photos of what you should have seen. The elodea is first, that's for part one. The cheek cells are second, that's for part two of the lab. Make sure you're answering these questions here. There's one question after each set of drawings. Once you've answered those, there's a couple of cell pictures for you to just label the parts. And if you look in your science book on page 52 and 53, that'll help you out if you need some assistance. And then this think question involves us putting a ruler under the microscope, and we didn't get a chance to do that in class. So I told you I'd have a video that would show you how to do this. So over on the right-hand side, I've got a couple of photos that show a clear ruler underneath a microscope. The first one is under the scanning power, so that's only 40 times magnified. And then the second one is under low power, which is 100 times magnified, which reminder that the low power is actually the medium-sized lens, because it's confusing. So over here on this think question, we're trying to get an idea of how big cells actually are. So we're going to follow these steps. If you were to put a ruler under the microscope, you would see what I have over on the right-hand side here. The small marks are millimeters. Those are going to be the smallest lines on a ruler if you're using the metric side. So you would see under scanning power, we can see 1, 2, 3, 4, like 5 millimeters. And under low power, if you look over here, we can see 1, 2, like maybe 2.5 millimeters is all. So that's because we're zoomed in 100 times. So how far across, the first question here is how far across the circle is it at 100 times? So if you look at this picture, you're going to be able to answer that. I also just said it. Then if you were to change it to high power, you would only be able to see 0.38 millimeters, which would be less than the spacing between these lines. So using that information, we're going to determine how big the cells you looked at yesterday actually were. So you're going to take the number of cells that you saw up above and you wrote down on those two questions. So that would be this question, how many elodea cells, the long way does it take to go across the lighted circle? And then you've got the same question about cheek cells. Use that number that you wrote down, and you're going to take that number divided by 0.38, and you're going to write that answer here. Okay, that's how you calculate it. So you take your number of cells across the circle divided by 0.38, and that's going to tell you the length of each cell. All right, hopefully that's helpful. And if you have questions, let me know. Have a great day.